Hi everyone, this is the third part of the transport across membrane lecture. In this third video, we're going to talk about facilitated diffusion and active transport. Starting out with facilitated diffusion, transport proteins can help facilitate diffusion across membranes for molecules that are not able to cross the membrane easily on their own. Hydrophobic substances easily diffuse across the cell membrane, like we've already talked about, and that's due to the composition of the phospholipid bilayer of the membrane. Just as a quick reminder down here, I know we've gone over this a few times. This is an image of the phospholipid bilayer. Remember, each phospholipid has the hydrophilic or the water-loving head and the hydrophobic or water-fearing tail. The membrane is structured so that the water-loving heads face outward towards the aqueous solutions on either side of the membrane, and the water-fearing or hydrophobic tails are what make up the interior of the membrane. Because the interior of the membrane is made up of those hydrophobic tails, other hydrophobic substances are easily able to diffuse across the membrane because they don't have a problem crossing that hydrophobic interior. However, polar or charged substances and larger molecules do not easily cross cell membranes. And that is because they have a hard time getting through the hydrophobic interior of the membrane. Water is a polar molecule, as we've already learned, but it is very small. So water molecules are able to very slowly work their way through the membrane. However, if you need water molecules to go through the membrane quickly, you need a transport protein. So since these polar or charged molecules can't get across the membrane easily or at all if they're too large, they move across membranes with the help of specific transport proteins, which is called facilitated diffusion. Facilitated diffusion is a type of diffusion, so it does not require energy, and it still relies on the concentration gradient. So molecules are diffusing from high concentration to low concentration. The only difference is that they are diffusing through a transport protein. Transport proteins are always specific for their specific solute molecule. So this transport protein here is only going to allow the yellow circle solute molecule to pass through. The red triangles will not be able to pass through this transport protein. I'm going to go out to our little whiteboard here. I made some changes from what you saw in the last video, and I added facilitated diffusion here. It is still a type of passive transport, so the passive transport rules apply. Molecules move from high to low concentration, and no energy is required. The difference in facilitated diffusion is that a transport protein is needed. So I'm going to write transport protein here. In facilitated diffusion, molecules diffuse down their concentration gradient through a transport protein in the membrane. Transport proteins help specific substances diffuse across the membrane down their concentration gradients and thus require no input of energy. The very rapid diffusion of water into and out of certain cells is made possible by a protein channel, a transport protein, called an aquaporin. This is an aquaporin here in the plasma membrane. Like I mentioned before, water is polar, but it is very small, so it can slowly work its way through the plasma membrane. However, if large quantities of water need to be moving into and out of the cell, you need a transport protein to facilitate this. And that's what aquaporins are. They're a channel that allows water to rapidly move into or out of the cell. A quick checkpoint question on facilitated diffusion. How do transport proteins contribute to a membrane's selective permeability? Think about that for a second and then we'll come back together. So if you said because they are specific for the solutes they transport, the numbers and kinds of transport proteins affect a membrane's permeability to various solutes, aka whatever transport proteins the membrane has are what is going to be allowed into the cell, since each transport protein is specific to its own type of solute molecule. Now we're going to talk more about the aquaporins that we just talked about, the facilitated diffusion transport proteins that allow rapid diffusion of water into cells. These aquaporins were actually discovered when someone was studying something completely different. So the research on another membrane protein, which was the Rh protein, led to the discovery of aquaporins. And Dr. Peter Agre received the 2003 Nobel Prize in Chemistry for his discovery of these aquaporins. He was not intending to discover the aquaporins. For a while, scientists had known that there was likely some type of protein that allowed for the rapid diffusion of water in certain types of cells, but they couldn't figure out what protein it was or where it was in the cells. They did notice that red blood cells and some kidney cells were able to rapidly gain or lose water. So they knew there was a transport protein that allowed this. They just couldn't find it. Dr. Agre was researching the Rh protein, which is used in blood typing for a positive or a negative blood type. He was researching it because it can be an issue if a mother has a different Rh protein than her baby when she's pregnant. So that's why he was researching it. And he had isolated two types of proteins. One was the Rh protein. The other was a smaller protein that he believed was a part of the Rh protein. But when they fluorescently tagged them, they realized that the smaller protein was not part of the Rh protein at all. And upon examining other types of cells, they realized that this smaller protein was found in really high abundance in red blood cells and in kidney cells. This then led him to think, and he talked to some colleagues who suggested that he could have possibly found the elusive water transport protein. He then did some studies, which this graph shows the results of those studies with frog eggs. He chose frog eggs because frog eggs, when they're incubating, they are in hypotonic pond water, which means they're pretty impermeable to water because they don't burst in the pond water. So they're not water can't leave or enter the cells very easily. They're impermeable to water. What he did, it was a really cool experiment. He isolated the DNA sequence for that small protein that he found that he thought was the water channel. He then got the mRNA sequence from that DNA and injected the mRNA into frog eggs. So he injected the mRNA that would code for the protein that they believed was the water channel. He then had a control group of eggs that were just injected with a buffer. It didn't have any RNA. He then put the eggs in water and measured what happened to them. The x-axis on this is the time in minutes and the y-axis is the relative volume of the eggs. We can see that the control eggs that were just injected with a buffer, their volume increased slightly at the beginning, but then stayed relatively constant. And even after an hour, it was about a 0.5% increase in volume, and they did not rupture. However, the eggs that he had injected the RNA coding for that water transport protein burst in less than 3 minutes. So they rapidly gained volume and then they burst before the 3-minute mark. This led Dr. Agre to conclude that that protein he was studying was the elusive water transport protein because it allowed water to enter and eventually rupture the previously impermeable frog egg. So a checkpoint question on this. 1, why were frog eggs such a good choice to test the function of this unknown protein? And then 2, why did they also monitor the behavior of control eggs in the hypotonic solution? So this is getting back to some experimental design that we talked about in our very first modules. So for question 1, since the frog eggs are quite impermeable to water, they're a good thing to test this on because in their natural state, they're not going to allow water in or not going to allow much water in. And so by seeing that a large amount of water was allowed in and the cells eventually burst, that meant that that protein was causing a change. And then for 2, they had to monitor control eggs to confirm that the frog eggs were actually mostly impermeable to water and to also compare the experimental results. So they provided a comparison to the bursting of the eggs that were making the aquaporin proteins because they themselves did not burst. So that's kind of a cool story about how aquaporins were discovered. And aquaporins are a type of transport protein that are key for facilitated diffusion to occur. The last main type of transport that we're going to talk about is active transport. In active transport, a cell has to actually expend energy to move a solute against its concentration gradient. So instead of going from high to low like passive transport does, active transport goes from low to high. And since we're moving molecules against their concentration gradient, energy input is required. Typically, the energy molecule ATP supplies the energy for most active transports that are carried out in the cell. We're going to see a few figures that show us the 4 main stages of active transport, but I wanted you guys to remember back to the question I asked you at the beginning of the transport section of this lecture about getting into a crowded club versus getting into an empty club with no line. We agreed that getting into a crowded club with a line out the door would be a lot harder than getting into an empty club. This is what active transport is. Active transport is like trying to get into a crowded club. The molecule is trying to move into a cell where there is already a high concentration of that molecule, or move outside of a cell where there's already a high concentration of that molecule outside the cell. So in order to do this, energy has to be used. Or if you think back to the club example, you could think of it as you're going to have to pay a cover charge to get into the very crowded club. I'm going to back out of this and go to our whiteboard and just write this active transport. So active transport, I'll write it in the blue color, is from low to high concentration. And because it is moving molecules against their concentration gradient, energy is required. So low to high concentration, and then I'll just say energy needed. And the energy that is typically used is ATP. So I'll write that out. ATP needed. So hopefully you can easily see the 2 differences between passive and active transport. Passive transport, high to low concentration, no energy needed. Active transport is low to high concentration, and so energy in the form of ATP is needed. Back to our PowerPoint. These next few figures show how active transport occurs in a cell. So we have the plasma membrane here and then a transport protein. This is the interior of the cell. This is the exterior of the cell. The first step is that a solute is going to bind to a transport protein. Notice this transport protein has a square spot that is perfect for these green squares to bind. ATP then will provide the energy for a slight change in the protein shape. Notice it closes this end and opens up this end and kind of kicks those green solute squares out onto the other side of the membrane. ATP provides that energy for this shape change. And then finally, the protein will return to its original shape where more of the green solute can bind. And again, this is active transport because we can see this one's less extreme of an example, but the concentration of green squares is higher on this side of the membrane than on this side of the membrane. So it's moving from low to high concentration. This animation provides a better visual image for how active transport occurs.
The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is co-transport. Here, both sodium ions and glucose move into the cell through a co-transporter protein. Sodium ions move down the concentration gradient created by the sodium-potassium pump, and glucose moves against its concentration gradient.
So those are two real-world examples of active transport. We're not going to talk much about the sodium-potassium pump in this class, but if you go further in biology and take cell biology, you definitely will talk a lot more about the sodium-potassium pump and why it's important. So a checkpoint question on active transport. Cells actively transport Ca2+ out of the cell. Is calcium more concentrated inside or outside of the cell? And explain. So take a second and think about that. If you said that calcium is more concentrated outside of the cell, you would be correct, because that active transport is pumping calcium against its concentration gradient, which is kicking it outside of the cell. Two other types of cell transport are exocytosis and endocytosis. All the transport we've talked about till this point is for relatively small molecules. Exocytosis and endocytosis are for large molecules, such as like the insulin protein that we talked about being produced in the biomolecules lecture, or a really large polysaccharide. All of those would have to be transported by exocytosis or endocytosis. A cell uses two mechanisms to move large molecules across membranes. The first is exocytosis. This is used to export bulky molecules such as proteins or polysaccharides. Again, we talked about how insulin is produced and then packaged through the ER and the Golgi apparatus and then secreted from the cell. It would be secreted from the cell through exocytosis because it is a bulky molecule. Endocytosis, endo just means inside, is the opposite of exocytosis, and it's when a cell takes in large molecules instead of sending them out. In both cases, exocytosis and endocytosis, the material to be transported is packaged within a vesicle that fuses with the membrane and either sends its contents outside of the cell or packs its contents and brings them inside of the cell. This video is a brief intro to exocytosis and endocytosis, and then we'll look at videos for each one of them.
Now let's move to the other side of our intestinal cell. Materials can be exported in vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell.
So I really like this video because it provides a good visual representation of those two processes. It clearly shows exocytosis exporting molecules from the cell and then endocytosis forming a vesicle that then goes into the cell. This example here is specifically exocytosis.
Exocytosis is the movement of intracellular vesicles to the plasma membrane, where they fuse with the membrane and release their contents into the surrounding fluid. This process occurs predominantly in secretory cells, such as mucus-producing cells or pancreatic cells, that secrete enzymes into the digestive tract.
We talked about how the pancreas produces insulin and secretes that. So this is how that is secreted from the cell, how that protein is secreted or the hormones are secreted from those cells. There are two types of endocytosis. So we talked about exocytosis, which is exporting from the cell. Now we're going to refer to endocytosis, which is bringing stuff into the cell. The two types of endocytosis are phagocytosis and then receptor-mediated endocytosis. In both of these cases, material is transported packaged within a vesicle that fuses with the membrane of the cell. Phagocytosis is the engulfment of a particle by the cell wrapping a cell membrane around it, which forms a vacuole. You can see this happening here. The cell stretches little appendages of plasma membrane called pseudopodiums out around the food or other particle and then completely envelops the particle, forming a vacuole, which then pinches inside of the cell. In certain protists that are single-celled, this is how they eat. In other types of cells, such as white blood cells that are in our body, this is how they attack pathogens or bacteria that are within the body. It's phagocytosis. So instead of this being food, it would be a foreign virus or bacteria that the white blood cell is engulfing through phagocytosis. And here's a video on phagocytosis.
In endocytosis, membranes invaginate or pinch in to form a vesicle, moving the enclosed materials inside the cell. This process can take different forms, each involving its own specific cell machinery. In phagocytosis, or cell eating, the cell engulfs debris, bacteria, or other sizable objects. Phagocytosis occurs in specialized cells called phagocytes, which include macrophages, neutrophils, and other white blood cells. Invagination produces a vacuole, which usually fuses with one or more lysosomes containing hydrolytic enzymes. Materials in the vacuole are broken down by these enzymes and degraded.
So a lot of the cell types she mentioned in this video are cell types that are related to our immune system. So they're how we combat foreign pathogens that are within our body, such as bacteria and viruses. The other type of endocytosis is receptor-mediated endocytosis. Receptor-mediated endocytosis uses membrane receptors for specific solutes. So the membrane receptors bind to specific solutes based on their shape. Notice that all these membrane receptors have a Y shape, which is perfect for the purple triangle molecule. So the Ys bind to the purple triangles, and then that membrane will invaginate or pinch in and form a vesicle of the whatever molecule they attracted with those receptors. Here's a video on receptor-mediated endocytosis.
Unlike phagocytosis and pinocytosis, receptor-mediated endocytosis is very specific. It is triggered when membrane receptors bind to specific external molecules, such as protein cholesterol complexes or proteins bound to iron. Membrane vesicles pinch off, and the external protein and its cargo are brought into the cell.
So as you saw in that video and in this figure here, sometimes you get random other molecules that were not specifically targeted. The purple triangles were the ones that were specifically targeted because those are the ones that the cells have the receptors for, but you will get some byproducts, excuse me, of some other molecules as well. So just a reminder, phagocytosis and receptor-mediated endocytosis are both types of endocytosis. Exocytosis and endocytosis are transport for large molecules. The other types of transport we talked about are for molecules that are much smaller. Phagocytosis is kind of like cell eating. It's where the cell's going to engulf food or another particle, could even be a bacteria if it's a white blood cell, and it will pinch off and form a vacuole, which will then meet up with a lysosome and its digestive enzymes to digest whatever is inside the vacuole. Receptor-mediated endocytosis is very specific for a certain type of molecule that the receptors are shaped to attract. You will get some byproducts with it, but the point of it is to get certain molecules inside the cell. An overall review of the transport across membrane lecture, we talked about two main types of transport that weren't the transport for the large molecules. We have passive transport that doesn't require any energy, and active transport, which does require energy. The three types of passive transport are diffusion, facilitated diffusion, and osmosis. Diffusion and facilitated diffusion solutes are moving from a higher solute concentration to a lower solute concentration. In diffusion, those molecules just move directly through the membrane. In facilitated diffusion, those molecules move through a transport protein, but still from high to low concentration. In osmosis, we're looking at a higher free water concentration moving to a lower free water concentration. Osmosis is specifically the movement of water from high free water to low free water. And it can do this directly across the membrane or through transport proteins. Finally, active transport is moving solutes against their concentration gradient from low to high. This requires energy input in the form of ATP. So this is the last video I want to show. It's a general review of everything that we talked about in this lecture, and it also links up to our next topic, which is digestion and the digestive system. Membrane permeability and selectivity directly relates to digestion and absorption of nutrients. So it's important to understand the links between these two before we shift gears and start talking about digestion.
We get all our energy and organic molecules from food. Before we can use the molecules we eat, they have to enter our cells, starting with the cells lining the small intestine. Let's zoom in to the surface of a cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can dissolve in the lipid bilayer. Notice how they move down their concentration gradient, from where they are more concentrated to where they are less concentrated. This is an example of diffusion. Diffusion is a form of passive transport. It does not require energy from the cell. Most molecules can't cross the lipid bilayer. Here, the sugar fructose moves into intestinal cells by facilitated diffusion, moving down its concentration gradient through a transport protein. Facilitated diffusion doesn't require energy from the cell, so it's also a form of passive transport. Water crosses the plasma membrane by facilitated diffusion, or by diffusing across the lipid bilayer directly. The diffusion of water across a membrane is called osmosis. The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is co-transport. Here, both sodium ions and glucose move into the cell through a co-transporter protein. Sodium ions move down the concentration gradient created by the sodium-potassium pump, and glucose moves against its concentration gradient. Now let's move to the other side of our intestinal cell. Materials can be exported in vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. On this side of the cell, we can also see oxygen and carbon dioxide diffusing across the lipid bilayer. Cells use all these processes to get what we need.
So that video was a great summary and leads nicely into what we'll be talking about in the next module, which is membranes and how that relates to digestion. This lecture PowerPoint ends with a few concept check questions. We're not going to go through them in this video since it is a video, but definitely look through them on the PowerPoint and make sure you understand the answers that are given. I'll see you guys next week in Module 9.